Okay, welcome to the second narrated lecture video. Just to recap, hopefully you took some time and you watched the videos on how MHC class 1 and MHC class 2 receptors get loaded and what T cell types they recognize. Just to recap though, MHC class 1 receptors are on all nucleated cells. All nucleated cells have to constantly be declaring their quote-unquote selfness. And again, what I mean by selfness is each one of us are individuals, and my self is only of Aryan origin. Something that is self to me, of Aryan origin, of the instructor origin, is going to be foreign to you, or something that your immune system should not recognize. So that's what we mean by self. And MHC class 1 receptors, which are protein receptors, are on the surface of all nucleated cell displaying selfness, happy, healthy self to the immune system. The moment they stop displaying selfness to the immune system will destroy that cell, thinking that cell has been invaded by some kind of virus or something. So, MHC class 1 receptors are on all nucleated cells. They are loaded in the ER, in the endoplasmic reticulum, and they are loaded with self molecules. Once they are loaded with self molecules, the receptor is placed on the surface of any nucleated cell, and that any nucleated cell can come into contact with CD8, otherwise known as killer T cells. MHC class 2 receptors are on antigen-presenting cells only. They are loaded not in the ER, but they are loaded in a vesicle in the cytoplasm. They are loaded with foreign peptides, foreign bits of pathogen that has been broken up and destroyed by the antigen-presenting cell. Once they are loaded in that vesicle in the cytoplasm, they are then placed on the surface of that antigen-presenting cell, macrophages and dendrites, and they come into contact and form binding complexes with CD4 cells, otherwise known as TH cells. Okay, so where do they bind, or how does that binding work? That is all functional on something called the T cell receptor. T cell receptor, or the TCR. Now, remember when we were talking about humoral immunity, we were talking about the B cell receptor, and B cells make antibodies, and each antibody and each B cell receptor has the ability to recognize and bind something ever so slightly different from its neighbor, meaning that all antibodies have a certain conserved area where all antibodies are the same, and all antibodies have a variable region where all antibodies are different. T cell receptors, or TCRs, are very similar to that. T cells undergo a similar maturation process where they have some somatic recombination events. In each T cell receptors are not secreted, but they're loaded onto the phospholipid bilayer of the cell. And all T cell receptors are the same in this constant region right here. All T cell receptors are variable, or different, in the variable region right there. So we end up with T cells that have the ability to recognize and bind something ever so slightly different than its neighbor. And it's those T cell receptors, along with the CD receptors, that form these binding complexes with the MHC class molecules. Okay, let's go through this a little bit slowly. Let's start with the antigen-presenting cell. Antigen-presenting cells, APCs, dendrites and macrophages, are the only cells that have MHC class 2 molecules. So this is the MHC class 2 receptor loaded on the outside of this antigen-presenting cell. This T cell, otherwise known as a helper T cell, otherwise known as a CD4, because it is a T cell, it has a T cell receptor. So here's the T cell receptor. And because it's a helper T cell, otherwise known as a CD4 cell, it's called CD4 because it has a CD4 receptor. So when these two cells want to have a conversation, when these two cells want to talk about antigens and what this helper T cell should get excited about, these three receptors come and make a binding complex together. This antigen presenting cell presents this antigen to the helper T cell. If this is something that this helper T cell recognizes, then this helper T cell or this CD4 cell will become activated. It'll go on to activate B cells. In the rest of the body, we have all of those nucleated cells. If this is any nucleated cell, any nucleated cell. So let's just say it's a kidney cell. So this kidney cell, because it's an any nucleated cell, has an MHC class I receptor. So here's our MHC class I receptor. Now, under normal conditions, this kidney cell has to be displaying normal, happy, healthy kidney cell-ness, selfness, to the immune system. That CD8 cell is a part of the immune system. If that kidney cell displays self, happy self, healthy self, then everything will be good to go. If that kidney cell is infected with some kind of virus or something, then that is going to stimulate that CD8 cell to kill it. But this binding complex, to have that conversation, requires that MHC class I molecule. And because it's a T cell, we have a T cell receptor. All T cells have T cell receptors. And because it's a CD8 cell, otherwise known as a killer T cell, otherwise known as a TC cell or cytotoxic T cell, it has a CD8 receptor. And all three of these receptors are required to make a binding complex for these two cells to have a conversation, for that CD8 cell to check the healthfulness of that kidney cell. So, like I was saying, T cells develop in the thymus. They go through a very similar maturation process that B cells do. Anything that doesn't bind appropriately to either MHC class I or MHC class II, depending on what kind of T cell it is going to be stimulated to undergo apoptosis. It's these T cells and these MHC receptors and their specificity. It's these guys that are in charge, or what happens when we get tissue transplant rejection or organ transplant rejection. Unfortunately, these MHC class I receptors and these mature T cells can be real troublesome. So another reminder, we have TC cells, otherwise known as cytotoxic T cells, otherwise known as killer T cells, otherwise known as CD8 T cells because they have the CD8 receptor. The job of these cells is to kind of peruse around, checking the healthfulness, making sure all of those nucleated cells are displaying healthy selfness. If they don't, if they're infected with a virus, then the cells will cease displaying self and display virus instead. If that T cell recognizes that it's not displaying self anymore, that killer T cell or CD8 cell is going to kill the virus-infected cell. The helper T cells, all helper T cells are otherwise known as TH cells, otherwise known as CD4 cells, because all helper T cells have the CD4 receptor on their surface. But we kind of break down the helper T cells into subgroups based on what they do, what their jobs are. So far we've been talking about helper 2 T cells. Helper 2 T cells are the ones that signal B cells to become activated and to clonally select themselves. Th1 cells basically signal antigen-presenting cells like macrophages to really become hyperactive and really kill a lot of pathogens that they're carrying around. So let's look at the cytotoxic T cells real quick. These are killer T cells. They kind of come into contact and move around, checking the healthfulness of each one of your cells, making sure that they are displaying happy, healthy self. If they're not displaying happy, healthy self, then much like those natural killer cells, they're going to basically vomit up toxins all over that potentially infected cell, and that infected cell is going to undergo apoptosis. Apoptosis is programmed cell death. It's basically where it causes the cell to commit suicide. And this is kind of what happens. This would be a CD8 cell coming into contact with cells. If it detects that the cell is infected with a virus, it's going to stimulate that cell to undergo apoptosis. It's going to cleave up the DNA and basically be destroyed. As that T cell then moves along to the next cell over, if it recognizes that that cell is infected with a virus, it's going to signal that cell to undergo apoptosis and just keep on moving on. At some point, we hope that it reaches non-infected cells, and when those non-infected cells are displaying on their MHC molecules happy, healthy self again, then it won't destroy it. Th1 cells are the ones that we test for. If you've ever had a tuberculosis test, a TB test, you know how they squirt, they take a little needle and a little bit of antigen and squirt it under your skin? That's actually to test whether or not you have activated macrophages and activated Th1 cells to tuberculosis. So they're not actually giving you tuberculosis. However, they're putting a little bit of tuberculin antigen under your skin. If you have Th1 memory, then at some point you've been exposed to tuberculosis. Now, that could be that you actually came into contact with an individual that had tuberculosis and you've been exposed to the organism, or since it's just looking for immunity against tuberculosis, then it could be that you've been vaccinated for tuberculosis, and that's going to cause a positive skin test as well. This is what it would look like if we got positive Th1 cell production. And then lastly, our other helper T cell, our Th2 cells, we've talked about them before. These are the ones that stimulate B cells to become clonally selected and get them to morph into plasma cells and secrete loads and loads and loads and loads of antibodies. Well, that's the end of cellular immunity. We will have a quiz on this, I am sure. Enjoy.